Yes, ma'am, no problem. Thank
All
you so
right.
much, ma'am.
Okay. Okay. Hello, am I audible?
Hi, ma'am.
Ayan, is that you?
Hello, hello, hello. Can you hear
Yeah.
me?
Is that you? Yeah, I can hear you.
Yeah, yeah, it is.
All right. Can you see my screen? Are you able to see my screen
No,
now?
no,
You can't see it now also?
I can't.
Okay, can you see it now?
It just says Mariam has this showing her screen. Their screen.
Okay, wait. What about now? Can you see it now?
Yeah,
All right. So, did you do your homework?
yeah. The one in the website.
Yeah, the one that had questions related to the chapter that we were studying. Did you do the homework?
Mm,
Any problems?
No. Could
All
you
right.
increase your volume a bit, just like your
I
voice?
have the mic in my hand only. Are you able to hear me now?
Yeah, I guess. Yeah, it's good. It's good.
All right. So, now we'll talk
DNA,
about, yeah,
not base pairing. We're doing base pairing, right?
yeah. We were talking about the DNA, and in the DNA, we saw that the DNA is you can say it is a nucleic acid, and each DNA molecule is made up of thousands of nucleotides. And now the nucleotide contains three things: the phosphate group, the sugar, and the base. Now we're talking about that base. Now, there are four types of bases that are present: thymine, adenine, cytosine, and guanine. So each pair of bases is held together by weak bonds, and there are four different base pairs in DNA: thymine, adenine, cytosine, and guanine. Now, adenine is denoted by A, thymine is denoted by T, and they will always pair up together with two hydrogen bonds. Same way, guanine is denoted by G and cytosine is denoted by C, and they will always pair up with three hydrogen bonds. If you reverse the situation, if you write T, then also it will pair up with A, and if you write C here, then also it will pair up with G. So this is the pairing, and this is how it will happen forever. Clear? So in the DNA molecule, in the double helix, these structures that you're seeing, these are the base pairing, wherein A, T, G, C, all the kinds of base pairings can be present. Any doubts up until now?
No. So
Or
they're all A is always paired with T.
A will always pair up with T and never with anyone else in DNA.
So same for all of them.
G will always pair up with C.
So it's same for all of them.
Yeah. But remember that in between A and T, there are two hydrogen bonds, and in between G and C, there are three hydrogen bonds. Clear?
Okay.
Any doubts up until now?
No.
All right, great. So now see, the two base pairs, whether it is adenine, whether it is thymine, guanine, or cytosine, they are joined together with the help of nitrogen, hydrogen bonds. And hydrogen bonds are weak bonds. Okay, those are weak bonds. Now, and what they do is that they keep the DNA double helix in shape. It gives the helical structure to the DNA molecule. Okay. So with this, we have actually completed. Now see, let us look at these key points. Also, yes, another thing. The double helix structure of the DNA, it was given by James Watson and Francis Crick. So if anyone asks you like who gave the double helix structure of a DNA, So Watson and Crick gave the double helical structure of the DNA molecule. Clear? Also,
Yeah.
did you do this? This part?
Yeah. Figure. Yeah.
Tell me. What will come after C here?
G.
Then here?
B. A will be with T.
Here?
G will be again with G will be with C. And G will be again with C. C will be with G. T will be with A. C will be with G. And A will be with T. And T will be with A.
This is forever. This will not change no matter what happens. Okay? Now,
Okay.
let us look at the certain key points. The DNA molecule consists of two strands coiled together to form a double helix. Clear? Each strand
Yeah.
contains the chemical bases adenine, cytosine, guanine, and thymine, which bond together. A always bonds with T, C always bonds with G, and vice versa. A chromosome is made up of a long supercoiled strand of DNA. The gene is the length of the DNA that codes for a production of a particular protein, and you can call the gene as the functional unit of DNA. It is the functional unit of DNA. So with this, we have completed the chapter that is DNA. I mean, this biological molecules. Now the next
Yeah.
chapter there is the enzymes. Shall we start the next chapter?
Yeah.
All right. Now in the enzymes, the first thing that there is to see is the structure and the action of enzymes. Now you might remember in the previous class, I did tell you that enzymes are responsible for increasing the rate of the reaction. Do you remember that? I told this to you in the previous class. See, a catalyst speeds up a chemical reaction and remains unchanged at the end of the reaction. Enzymes are proteins produced by organisms that speed up chemical reactions. So they are known as biological catalysts. Do you remember me telling you this thing about the enzymes? Yes or no?
Can you hear me?
Yes, I can hear you. Can you hear me, Ayan?
Yeah, I can.
All right. Okay. So yeah, we were talking about what biocatalysts were. Now I was asking you the question as to, do you remember me telling you this thing that there are certain molecules that can increase the rate of the reaction, and those are known as the catalysts, and the enzymes are the biocatalysts. Do you remember me saying this?
Just now?
No?
Yeah.
Anyway, even if you don't remember me saying this, I'm saying it now. The enzymes are proteins that are produced by organisms and they speed up the chemical reaction, and they are known as biological catalysts. So the other name for enzyme is biocatalyst or biological catalyst. It's the same thing. Now, how enzymes work? Many chemical reactions take place in organisms. These reactions happen too slowly to keep organisms alive unless they are speeded up by the enzymes. There are many different types of enzymes as each one catalyzes a different reaction. Another very important thing to remember about enzymes, that enzymes are very specific. Enzymes are extremely specific, meaning they will only react to the kind of reaction that they are supposed to and not to any other. So they are like, for every reaction, there is a different enzyme. Most enzymes work inside cells, but many of those that we will discuss here work outside cells, for example, in the gut. So here the enzymes that we are going to discuss in this chapter, they work outside the cell, but maximum of the enzymes, they work inside the cell. Clear?
Yeah.
All right. Now, catalyst is a substance that
Yeah.
speeds up a chemical reaction and it is not changed by the reaction, meaning at the end of reaction, product will be formed, but the catalyst will stay the same. Enzymes are proteinaceous and they act as biological catalysts to speed up the rate of reaction. Now, the enzymes, they follow a lock and key method. We'll discuss about it later on. Now, let us see the reactions that the enzymes catalyze. It is three types. The first is breaking large molecules into small ones. This is important in nutrition when large food molecules are broken down into small ones so that they can be absorbed and then used. Bacteria and fungi releases enzymes to break down their food, and we release enzymes in the gut for the same reason. So there are some enzymes that are responsible for breaking down larger molecules into smaller ones. And since we are talking about the gut where the digestion takes place, so here we are discussing that larger molecules will be broken down into smaller. For example, if this is the size of the molecule, after the enzyme acts on it, it will be broken down into smaller pieces so that it is easy for the enzyme to digest. Here,
Yeah.
the second is the building up of large molecules from small ones.
Wait,
Now,
could you repeat that part again? The part where enzymes work inside cells.
there are like most of the enzymes that are there, they work inside the cell, but there are
Okay.
certain enzymes. Now in this chapter, whatever enzymes that we are going to study about, they work outside the cell, meaning they work inside
Okay.
any organ system.
Okay.
Now the second type of you can say enzyme catalysis is the building up of large molecules from small one. The first one was from large molecule, it can be broken down into small ones, but here the small ones are being used to make the large molecule. Now, small molecules such as glucose, they are joined together to make large molecules. These enzymes work inside cells to speed up formation of storage molecules such as starch and structural molecules such as cellulose for cell wall of plants. So we know
Okay.
that when many glucose molecules come together, they form starch and cellulose. And
Yeah.
this process of forming the starch and cellulose, this also takes place by the help of the enzyme. So here, from smaller molecules, larger molecules are being made. Clear?
Okay.
And the third way by which the enzymes act is converting the small molecule into another molecule. Many of the chemical reactions that occur inside cells involve small changes to the molecule, such as adding or removing atoms or groups of atoms. For example, there are enzymes that remove hydrogen from compounds during respiration. So here, the enzyme is responsible for changing the molecule and the product that is formed is completely new thing. So this is how it happens. The substrate is the structure on which the enzyme will act upon. So this substrate and goes to this site which is known as active site. Every enzyme has
Can you give me a
active.
second? Give me- Okay, teacher, you can continue from the enzymes and substrates.
Yeah, so as I was saying, the substrate is this, you can say it is the molecule on which the enzyme will act upon. And now for this process, the enzyme has certain sites that are present in it. They are known as active sites. Active sites are those parts of the enzyme to which the substrate will come and bind to. So this substrate now goes and binds to this active site. And then the enzyme
Okay.
acts on the substrate and breaks it, or it forms the products. So this is how the enzyme action happens. Clear? Always
Hmm.
remember that enzymes, they have active sites present. These active sites are
So
the places where the substrate will come and bind to.
after binding, the product gets released from the enzyme.
Hmm. And the enzyme has no changes in it. The enzyme remains unchanged. Clear?
Yeah.
Now the enzymes are made up of protein. All enzymes have 5 important properties. They are all proteins, meaning all the enzymes are proteinaceous. Each enzyme catalyzes one reaction. So they will catalyze only one reaction because they are specific. They are specific. They can be used again and again because they are not used, or you can say they are not changed during the chemical reaction. Now, enzymes, they are influenced by the temperature and they are influenced by the pH. So their working will vary with the changes in the temperature and the changes in the pH. Okay.
Okay.
Enzymes are made up of protein molecules. These molecules can be folded into many different shapes. Each type of enzyme molecule has a shape that makes it suitable for catalyzing one type of reaction. This explains why there are many different enzymes, one enzyme for each reaction. There are no two reactions that are done by the same enzyme. Every reaction has a different enzyme of its own. Now, the enzyme, it catalyzes reactions in which substrates are converted into products. Now, if you see here, for example, the active site is here and this is the substrate. Now, two substrates are going there and being present or getting binded at the active site. Now, the enzyme is acting on the substrate and it is joining it together, and then at the end, it releases the joined molecule. So, the enzyme can break the molecule or it can join the molecule. Notice that the shape of part of the enzyme matches the shape of the substrate molecule. Why? Because it is specific. The enzyme and the substrate have shapes that are complementary, so they fit together. Since the enzyme and substrate relationship is so specific, so the enzyme will have, both of them will have shapes that are complementary, so that one will go and fit into each other. Once they fit together, the reaction can take place. Other substrates have the wrong shape to fit into the enzyme, so it will not involve in the reaction catalyzed with this enzyme. Now, when the reaction is over, the product or the products leave the enzyme and another substrate molecule enters. But this enzyme remains unchanged. Nothing happens to the enzyme. Clear?
Okay.
Now see, if you look at here, this is the structure of the enzyme. And here the substrate A is there and substrate B is there. So only the substrate A will fit and the reaction will take place. But if the substrate B will come here, no reaction will take place because this is not complementary to each other. Clear?
Yeah.
Okay, just
Yeah.
1 second. All right, now as you can see in figure 5.1.2, this one, the part of the enzyme where the substrate fits and where the reaction take place is the active site. Active site is the part wherein the substrate will come and fit into. Now, this way of describing how enzyme work is known as the lock and
Which
key
figure?
model.
Figure 1.5.1.2, right? Or 5.1.3?
Both. You can take both of them. The place
Yeah.
where the substrate will come and fit into, it is known as the active site, as I told you before also. So this substrate will come and bind onto the active site.
They bind and then they are released as a product together.
It will bind and then the chemical reaction will take place and then the enzyme will release the product and be unchanged. Clear?
But
Same goes
the
for all the
product,
reactions. Hmm?
the product will be the substrate will they will bind together, right, as one product.
Yeah, in this particular one, two substrates are coming, and since their shape and organization is such a way that they can fit inside the active site of the enzyme. So now the enzyme is
Yeah.
causing the joining of the two substrate and then releasing of one product. So this is when the molecule is being formed. The other is when the molecule is being released. It is being, you can say, differently broken down or stuff. Okay.
Okay.
Now, the way of describing how enzyme work is known as lock and key model. Just like how we know that there is like a specific key that will fit inside the lock and then the lock will open. Same is the action of the enzyme. There is a specific substrate which will fit onto it and then the reaction will take place. Clear? The enzyme and substrate combine like a key entering a lock to form an enzyme substrate complex. This part wherein the enzyme and the substrate is present together, this is known as enzyme substrate complex. Clear?
Yeah.
Now, let us see what are the key points. Enzymes are biological catalysts that increase the rate of chemical reactions. Enzymes
Enzyme,
catalyze.
but yeah, enzymes are proteins, and each enzyme acts as one type of reaction.
Yeah. Enzyme catalyze reactions in which substrate molecules are converted to product molecules either by building up or by breaking down. It can build up the product, it can break down the product. Now, all enzymes are protein. Part of the enzyme where the reaction occurs has a complementary shape to the substrate. And this part wherein the substrate fits is the active site of the enzyme. And this is where the reaction also takes place. Clear? These 4 points are clear to you?
Yeah.
So here enzymes are biological dash. What are they?
Catalysts.
Okay. That dash up the rate of chemical dash.
The dash speed up the rate of chemical reactions. No, chemical reactions. Yeah.
Enzymes are all dash
Proteins.
and each enzyme acts on one type of dash.
One type of substrate or one type of
Substrate.
reaction.
Substrate.
Substrate. Okay.
It acts on one type of substrate or it acts in a single type of reaction. Any doubts up until now? Just do one thing. Take 2 minutes and read this till here. And then if you have any doubts, we'll see it. If you don't, then we'll continue. Okay, just take 2 minutes or 5 minutes. You can read it nicely and tell me if you have any doubts.
Okay. So you want me to read it? Hello.
Yes, just read it on your own. You don't have to read it aloud. And till here, till up to where I've taught. I'm asking you to
The
read it
supplement,
so that,
right? The supplement.
yeah, till here, the supplement part. Meaning from the beginning of the chapter till here, you read it. If you find any difficulty, you ask me. If you don't find any difficulty, then we'll move on to the next topic.
So the supplement wants me to answer the question why each enzyme will act on only one substrate, right?
Yes, each enzyme
Because,
will have its own
because that
substrate.
enzyme is only complementary to that substrate.
Exactly.
Then next one is why an enzyme can be used to catalyze a large quantity of substrate.
Yes, because it speeds up the rate of reaction.
Oh, so because they bind with more than one substrate?
No, it doesn't bind with more than one substrate. Rather than that, it like binds with one substrate, forms a
More
product.
substrate to
Yeah,
repeat the catalyst.
yeah. Because once it binds with one substrate, then forms the product, then again with another substrate, then forms the product, and so on.
Last one is why destroying the active site by heating the enzyme to a high temperature stops the enzyme from working. Because the enzyme will lose its complementary
Exactly.
uh, shape.
Meaning the substrate will not have any place in the enzyme to bind to. And when the substrate cannot bind, then how will the reaction take place?
Enzymes are biological catalysts that increase the rate of chemical reaction. Enzyme catalyst reactions. Can you hear me, ma'am?
Yes, I can hear you.
Okay.
Yeah, I have asked
So
you
I
to
read
read
them.
it. The whole
Yeah, I
thing
did.
you've read
So
so quickly.
the three key points, right? I mean four.
No. I am asking you to read the entire thing till the end. That point we have taught till, like here.
Oh, you want me to?
Yeah, from the
Okay. From a catalyst speeds up a chemical reaction till the supplement.
yes. Till there and find out if you have any doubts. If you do, I will solve those doubts. If you don't, then we'll carry on. And
How about explain the second point, building up larger molecules from smaller ones?
okay. So they have given here that the enzyme can act by three ways. Either it can break one large molecule, it can build up one large molecule from smaller molecules, and then it can convert one small molecule into something another. These are the three ways by which the enzymes can act. Clear? Yes, are you saying something?
Can you hear me?
Yes, I can hear you.
Could you repeat it again? The second point, building up molecules from small ones, larger molecules.
Yeah, just like how there are many tiny molecules that when they join together, they form one large molecule. Just like how many glucose molecules they join together to form the cellulose or the starch. So just like that, this whole entire thing wherein the small molecules they join together and they form the large molecules, that is how the enzyme works. Either the enzyme
So
will
the
break.
enzymes bind them, right? They bind them. They bind these small
Yes. Yes.
substrate.
Yes.
This speeds up the storage of molecules from
It speeds up the formation of the storage molecules. The formation.
from
The
the
enzyme will
storage
speed up the
molecules are the
reaction
product.
rate. Yes.
So the storage molecules are the
Yes.
product,
Yes.
and substrate are the storage molecules.
Hmm?
So
No.
substrates
Here,
are
substrates are the ones on which the enzyme will act upon. They are not the storage molecules. They have given the examples of starch and cellulose. Starch is the storage molecule of plant. We know that it is the stored food product of the plant. So they have
yeah,
given this
yeah.
as an example. This is not the substrate. The substrates are not storage molecules. The substrates are those
Substrates are
substances on which the enzymes act upon.
so all okay. Small molecules are completely joined together. Many of the chemical reaction. So in the even the figure, the process starts from the product release and it fits into it binds with the active site.
What?
So the first in figure 5.1.1, the first thing happens is the product is released and it is it
No. No. The first
the
thing will
released
happen is the
product, yeah,
substrate.
the release, this is the released
No.
product. First the
The released
substrate,
product. No, start it from here.
yeah, I know. The first the substrate binds with the enzyme, then
Yes.
there's the released product.
The products are released after being formed.
Are they released in two or just in one form?
Depends upon if the reaction is forming two products or three products. It depends upon the rate of reaction or the rate that the reaction, what it requires. For this reaction, two products are being released. So it means that they are being done into two pieces. But if it requires three, then three pieces would have been released.
Oh, so it'll produce the amount of pieces in um according to how much is needed.
Yes. Yeah, that depends on the different types of reaction.
Enzymes are made of proteins. All enzymes have five binding. So they're also influenced by pH.
What?
They're also influenced by pH.
Yeah,
So,
they are influenced by the pH, they are influenced by the temperature. We'll see how in the next
mm,
topic.
oh, in next topic, okay. So we will see how much its H plus atoms are or ion.
Yeah, like how does it gets influenced? Whether high heat, you can say, changes it, low heat changes. We'll see that in the next topic.
Okay.
Done?
Yeah, I'm done. Once you
Done?
fit together, yeah, you can take this. Yeah.
Any doubts?
No, I asked you already.
So the next topic under the chapter enzyme is the factors that affect the enzyme action. The first one is the temperature. The activity of an enzyme is determined by measuring the rate of reaction that the enzyme catalyzes. This may be done either by measuring how much product is being formed or by measuring how much substrate is being used over a period of time. The rate is like the speed of the reaction measured in quantity of product or substrate per unit time or per minute. For example, if we have provided 5 substrates and 5 products are being formed, then we can say that the enzyme is working perfectly. It's working just like how it is supposed to. Now let us see how.
If the measured amount of quantity is formed, if the
Hmm,
product fits product fits the quantity needed.
yeah, if more and more products are being made, it means that the enzyme is performing flawlessly. If less products are being made, then maybe some problem is there.
So how would you fix the problem?
How would we fix the problem? We'll fix by increasing the concentration of enzyme. That is far off from your topic. That does not come under your syllabus. We'll increase the concentration of enzyme, but that is not something you have to study. Now, effect of temperature on enzyme. The activity of enzyme is influenced by temperature. This graph shows the effect of increasing temperature on the rate of a enzyme catalyzed reaction. So when the temperature increases, we'll see how and what happens. This is the graph that shows that with the increase in temperature from 10 degrees Celsius to 60 degrees Celsius, what happens to the rate of the reaction or the how much product is being formed. Now, the rate of reaction is slow at low temperature, meaning at 10 degrees Celsius, the rate is very slow. As the temperature increases to 40 degrees Celsius, the rate of the reaction increases. It reaches a maximum at 40 degrees Celsius. Maximum at 40 degrees Celsius. And then it starts getting reduced if we increase the temperature above 40 degrees Celsius. So, and then it becomes 0 at 60 degrees Celsius. So we can say that the temperature is the condition on which the enzyme will work upon only when the temperature is optimum, meaning when it is what the enzyme requires. For example, if any enzyme, let's see, different enzymes work on different temperatures. So if there is an enzyme that works the best at 40 degrees Celsius, so below 40 degrees Celsius, it will form low products, and even above 40 degrees Celsius, it will form low products because it works at an optimum temperature. Optimum temperature is the exact temperature at which the reaction will happen or the enzyme will work on. So the temperature at which the maximum rate of reaction occurs is called as the optimum temperature. This is the best temperature for the enzyme. Then there are some examples of optimum temperature. The enzymes from the fungus and the plants, meaning fungal and the plant enzymes, they work at approximately 20 degrees Celsius. The human enzymes, they work at 37 degrees Celsius because this is the normal body temperature. And then there are certain enzymes that are produced by bacteria for use in industry. They can work at higher temperature as well. So different enzymes, they work at different temperature. So we can say that different enzymes have their own optimum temperature. Is this much clear to you?
It is, yeah.
Any doubts?
No.
All right. Now, an enzyme molecule is folded into a shape that accepts the substrate molecule. The shape of an enzyme's active site is maintained by bonds between different parts of the molecule. Remember that the shape of the active site determines whether the substrate will fit into the enzyme for the reaction to occur, because it is very specific and it has to be complementary in order for the reaction to take place. Now, at first,
Yeah.
increasing the temperature of an enzyme control reaction will increase the rate. This is because enzyme and substrate molecules have greater kinetic energy. So as we increase the temperature, the rate of the reaction will also increase. As we saw that on increasing the temperature up till 40 degrees Celsius, the rate of the reaction increase. Clear? Now,
Yeah.
they move around more quickly and there are more chances of them colliding, the substrate fitting into the active site and the reaction taking place. So as we increase the temperature, with this, the kinetic energy increases. Kinetic energy is with respect to the motion. So as the motion is increasing, now the substrate is coming quickly to bind to the enzyme or to bind to the active
Yeah.
site, which is causing the
Rate of
reaction to take place. But if the temperature is higher than the normal temperature, at a higher temperature, the bonds holding the enzyme molecule together, it starts to break. Because we know that the enzymes are protein, and protein, they start getting denatured at a higher temperature. Denaturation of
yeah.
enzymes takes place, or the protein. This changes the shape of the active site, so the substrate no longer fits. We say that the enzyme has been denatured and it no longer catalyzes the reaction. So at high temperature, the enzyme can get denatured. Clear? Whereas at low temperature, the enzyme gets inactivated. So the enzyme only works on the optimum temperature. Clear?
Yeah.
Now let us look at the key points. Increasing the temperature of an enzyme controlled reaction increases the rate of the reaction up to a maximum which occurs at the optimum temperature. Beyond this, the reaction will not take place. This is because greater kinetic energy is causing greater number of collisions. Collision means binding, or you can say the enzyme and the substrate, they are being collided. They are being, what do you say, like when two products, they smash into each other, or two substance, they are coming and they are joining together, so that is collision.
Binding.
Yeah, they're
Yeah.
binding, and that happens between the enzyme and the substrate. Now at higher temperature, the rate of the reaction will decrease until it stops acting like a catalyst. So at higher temperature, it doesn't mean that, okay, we were increasing the temperature, so the rate of the reaction was also increasing. Now when we increase it further, the rate of the reaction will not increase, because now the enzyme molecule will start getting destroyed or denatured. This is due to the change in the shape of the active site, which means that the substrate can no longer fit. The enzyme is now denatured. Clear?
Yeah. Denatured. Okay.
Any
So
doubts?
the
Hmm.
substrate cannot fit with the active site.
No, it won't.
Okay, I got it.
And then we'll talk about what is the relation between enzymes and pH. But before that, you read it once about the enzyme and temperature. The reason why I am asking you to read is, many a times you, like, students think that they've understood, but when they read the book, they find difficulties again. So that is why I'm asking you to read it here, so that you don't have any difficulties in the future. Okay? Yeah, so read it, like read from the beginning till then, and then we'll continue with the enzymes and pH.
Activity of the enzyme. reaction that is the enzyme. So how are we supposed to know the optimum temperature for an enzyme?
How are we supposed to know the optimum temperature of the enzyme? See,
Yeah,
there are like the maximum enzymes, they work at 40 degrees Celsius, maximum of them. But then
all of them.
again, maximum, not all, maximum of them. But then there
Okay.
are certain enzymes that have different optimum temperature, and that cannot be known as such. We have to see their working, then only we can understand. Meaning we have to perform this experiment wherein we'll decrease the temperature and see the rate, then we'll increase the temperature and we'll see the rate. So we have to do it for every enzyme in order to find out what is the optimum temperature for it. Done?
Most at high temperature. And this is due to change in sugar. Yeah, done.
Any doubts?
Science and pH. No.
See this thing here, they have written. You may be asked to describe the effect of temperature and pH on the enzymes, but only in paper 4 will you be asked to explain how these are affecting. For example, in longer answer questions, you'll be asked like how the temperature and pH affects the rate of the reaction, or the how does it affects the enzyme. So then you have to explain as to increasing the enzyme and what happens after decreasing the temperature and increasing the temperature. And remember that the enzymes are proteins. We have studied about proteins, right? Proteins are made up of what?
Proteins are, yeah.
Yeah, proteins are made up of what? They're made up of dash.
Uh, oh, for the last one. So we did proteins are made up of
I'm asking you,
amino
what are
acid,
proteins made up of? Yes,
amino acids, amino
amino
acids,
acids. Yes, amino acids.
amino
And
acids.
since enzymes are proteinaceous in nature, they get denatured by high temperature and at
So
high
the
pH.
proteins get denatured by high temperature, or proteins don't have that? Never mind, never mind.
Yeah, the protein, since proteins they get denatured at high temperature, so since enzyme is protein only, so this also gets denatured. Denatured means that its structure gets destroyed. If its structure gets destroyed, then the active site shape also changes, and if the shape of the active site changes, then no substrate can come and fit into it, so no rate of reaction will take place. Clear?
Uh-huh. Okay.
Yeah. And are you studying the chapter, like the done chapter side by side?
Yeah,
Because if you leave it, then it's of no use. You should do it
yeah,
side by side only so that it will be fresh. And you did not have any difficulty in biomolecules chapter?
no. I'll go through it one more time though.
Yeah, that is what I'm saying. Once the chapter is done, you're supposed to read it on your own, and you're supposed to learn, like start learning it. Because
Yeah,
you know it's my job to teach you, but then it's
yeah,
your job to study it on your own, right? So once the chapter is completed,
yeah.
learn every single thing that is there. Okay.
Okay.
So today we have discussed about what enzymes, like what enzyme structure is, how do the enzymes act, and what is, like, if you want to define the action of enzyme, what do you call that kind of condition as? Like, how will you explain the action of enzyme? By which example?
How will I explain the action of enzymes?
Hmm.
By using complementary and
No,
the
by lock and key model.
yeah, complementary and locking key. Both.
Lock and key. No,
Complementary.
lock and key model.
So I don't have to use
Compl-
complementary.
Lock and key uses that complementarity that you're talking about. Okay.
Uh,
So if we want to explain the action of the enzyme, the best way to explain it is by lock and key model. Just like how there is a specific key for every lock, same way there is a specific enzyme for every substrate. Enzyme has the active site and everything about it. Clear?
yeah.
All right. Now let's just keep it till here only because the time is up. In the next class, we'll start with enzymes and how they are influenced by the pH.